Okay. Assalamu alaikum, everyone. I'm Nour Bajawi, a second year medical student, and today I'll be explaining enzymes and enzyme kinetics. So we'll go over the enzymes, and we'll have an intro to the enzymes. Then we'll talk about what influences enzyme activities, the types of enzyme substrate complex, and then the enzyme kinetics, allosteric enzymes, and their regulation, and then lastly we will solve questions. So what are enzymes? Enzymes are proteins. They are biological catalysts. They can either be made from protein, or they can be RNA molecules such as ribozymes, which are used for RNA splicing. Enzymes can also be they are catalysts. They're chemical agents that increases the rate of the reaction by lowering the activation energy, and they do not change in shape. They're the same shape throughout the reaction. They do not change in shape. They are selective, which means that they are specific. The specificity of the enzyme depends on the 3D shape of the enzyme. It does not bind to any substrate. It is specific for its own. Each enzyme is specific for its own substrate, such as, for example, trypsin, which hydrolyzes the peptide bond. Okay. Then we have enzymes are recycled. They can be reused. They're not only used once, they can be reused multiple of times. So an enzyme consists of an active site. This is where the substrate bind. And this active site is very specific to its substrate, okay? And the active site, the shape and the chemical environment of the active site is what allows the chemical reaction to proceed. Then we have the substrates. The substrate, as I said, they are the reactants that are activated by the enzyme. They bind to the active site to produce an enzyme substrate complex, which will then form a product. So as we said, enzymes are catalysts. What do you mean by catalysts? Catalysts is that they increase the rate of the reaction by lowering the activation energy to form products, okay? They increase, by other means, they increase the favorable orientation of colliding reactant molecules to produce for the products to form, okay? So they increase the chances of the active site and the substrate to collide and join together to produce a product. So we said that they are a catalyst and they decrease the activation energy. What do we mean by they decrease the activation energy? They decrease the activation energy that is needed for the reaction to proceed. You can see in this graph, the purple graph is where we used the catalyst, and the blue graph is where we didn't use the catalyst. And you can see the difference between the activation energy here. The activation energy is needed for the reactant to reach a state called the transitional state. In the transitional state, this state is where we will it's like a peak of energy that is needed for the reaction to reach it, so that it can proceed to form the products. Okay. So as we said, the activation energy is the energy required for each for the substrate to reach the transitional state. It is the peak energy so that it can move into to form the products. Okay, and transitional state is the peak energy barrier that must be overcome for the reaction to proceed and to form products. Now we're going to talk about the lock and key hypothesis. What do you mean by lock and key hypothesis? It is where the substrate and the active site of the enzyme fit so perfectly together, okay, to produce an enzyme substrate complex. And then we produce product. The product obviously is going to have a different shape than the substrate. So we talked about the lock and key hypothesis. Now we're going to talk about the induced fit model, where the active site has a different shape than the substrate. But when the substrate binds to the active site, you can see the different shape in the substrate and the active site. But when the substrate binds to the active site changes its configuration, okay, and to accommodate the substrate and form an enzyme substrate complex, which will then form products and enzymes. So once we release the product, the active site, you can see that it went back to its normal shape, so that it can accommodate the other substrates to produce more products. This change in shape, as we said here, the change in shape of the substrate of the active site will cause the lowering of the activation energy, and this will cause an increase in the rate of the reaction. So we said that enzymes are specific. So enzymes may recognize and catalyze. So they're specific. So they're specific to three things. Either they can recognize a single substrate, which is only recognize this active site only recognizes one thing, such as urease, for example. This enzyme only catalyzes the hydrolysis of urea, does not catalyze the hydrolysis of any other thing. Okay, or a group of substrate where an example is hexokinase. Catalyzes the addition of phosphate groups to other hexoses. Okay, like to different type of hexoses does not differ, يعني does not say only one type of compound. No, all type of hexoses. And then we have a particular type of bond. They only catalyze a particular type of bond, such as, for example, chymotrypsin catalyzes the hydrolysis of a peptide bond, this type of bond, or like let's say ester bond, and so on. Then we have here the types of bonds. So we have six major important types of enzymes. So let's start with oxidoreductase. Oxidoreductase catalyzes the oxidation and the reduction reaction. So it's like a redox reaction. So you'll see here, for example, NADH NADH is going to be the oxidizing agent. The lactate will be reduced into pyruvate using the enzyme lactate dehydrogenase, and then NADH, so lactate is getting oxidized, and NADH is getting reduced using the enzyme lactate dehydrogenase. So it catalyzes a reaction where we have oxidation and reduction in them. Okay, then we have transferases. Transferases catalyzes the transfer of either a carbon, a nitrogen, or a phosphate containing groups, such as in this example where we have serine hydroxymethyl transferase enzyme catalyzes the transfer of this methyl group into tetrahydrofolate, okay, to form a glycine. And this tetrahydrofolate bonded to the methyl group, okay? Then we have the hydrolases. From its name, hydrolases means it involves water molecules, such as here catalyzing the cleavage of bonds by the addition of water. When urea is hydrolyzed using water to give us carbon dioxide and ammonia. So in hydrolases, we use water to break down the compound. In transferases, we transfer carbon to a carbon or a nitrogen or a phosphate to another compound. And in oxidoreductase, we reduce and oxidize in the same reaction. Then we have the lyases, which means that it catalyzes the cleavage of either a carbon-to-carbon bond, a carbon-to-sulfur bond, or a carbon-to-nitrogen bond, such as in the reaction where pyruvate is broken down, or it's broken down into acetaldehyde and a carbon dioxide. You can see that, like, take it from me, it's like one reactant and two products. So it's one reactant is broken down into two things. Okay. Then we have the isomerase, which catalyzes the racemization of the optical or geometric isomers. So in isomerase, here we only have a change in the configuration of the bonds in the compound. We're not adding or removing. We're only changing the configuration of the compound to form another compound. It's like isomers, so they are isomers. And in ligases, we add ligases. So catalyze the formation of bonds between carbon and oxygen, sulfur, nitrogen grouped, coupled to hydrolysis of high energy phosphates. So here we have the pyruvate. In the pyruvate, we added carbon to the pyruvate to form oxaloacetate. So we added them. So we joined them together. This is what ligases mean. Okay. So now we're going to talk about zymogen. What does zymogen mean? What do we mean by zymogen? A zymogen is an inactive enzyme. So it's an inactive enzyme that is released to the environment. So it is inactive and it is released as inactive enzyme, and then it is activated using a biochemical reaction. Okay, such as, for example, when pancreas releases the inactive pepsinogen into the environment, and then into the stomach, and then in the stomach, it will, when it interacts with the acidic environment, it will be activated to form a pepsin. Okay, why do we do this? Why do we not just release the pepsin itself, the activated enzyme? Because if we release the pepsin itself, the activated enzyme, what will happen is that this enzyme will start digesting the proteins in the cell and the pancreas, and it will lead to self digestion of the organ and damage. So we don't want this, so we just release it in its inactive state, and then when it reaches when it's there in the area, in the place that it's acting on, it will get activated by, let's say, for example, pepsin, the acidic environment. So we will now talk about what influences the enzyme activity. So what do you think influences enzyme activity? We have three things that influence enzyme activity. These are the environmental conditions, the cofactor and coenzymes, and enzyme inhibitors. So we'll talk first about the environmental conditions. What are the environmental conditions that might affect the enzyme's activity? These are the temperature, the pH, and the ionic concentration. How do these affect the enzyme activity? The temperature. So we know that as we increase the temperature, we increase the rate of the reaction, right? So there is something called an optimum temperature, where if we increase the temperature more than this optimum temperature, we will start denaturing the enzyme and destroying it. And now we're not going to increase the rate of the reaction. We will start to decrease the rate of the reaction, because we're going to destroy the enzymes, and we don't have enzymes to produce products. So we have something called the Q10 in the temperature. The Q10 measures the increase in the rate of the reaction every 10 Celsius rise in temperature. And chemical reaction normally have a Q10 of 2 to 3, which means that every 10 Celsius degree rise in temperature, the rate of the reaction either doubles or triples in amount. Okay. So as we said, in a very high temperature, denatures protein, and we will be denaturing enzymes and so on. So now we're going to talk about pH. The pH. So same thing for pH. The pH, we have an optimum pH. For each enzyme has an optimum pH. If we exceed this optimum pH, we will start to denature the enzyme and alter the active site configuration. When we alter the active site configuration, the substrate won't be able to bind to the active site, and thus we will decrease the enzyme activity and decrease the amount of enzymes we have for the substrates to react with. So we don't want to increase the pH because if we increase the pH, we're going to alter the enzyme. So we finished from pH, we finished from the temperature. Now we're going to talk about the enzyme factors, the coenzymes and cofactors. Coenzymes are non-protein molecules that help the enzyme's function. It's like coenzyme. It's needed for the enzyme to be there. So the enzyme alone itself is called an apoenzyme. It's an inactive form. Okay. When we add coenzyme to the enzyme, we will activate the enzyme. So the coenzyme, it is, as you see here, it is associated with part of the active site. So it is when it binds to the enzyme, it becomes an active enzyme and leaves some part for the substrates to bind to it. Okay. So we have different types of coenzymes. So we have here. So when we bind the coenzyme with when the coenzyme joins the active site, it will form something called a holoenzyme. So holoenzyme, here we have the apoenzyme, the inactive enzyme, binds to the coenzyme, such as, let's say, co-substrates. An example might be NAD, or vitamins. Okay, when they bind to an apoenzyme, they will form a loose bond. Okay, while for prosthetic groups, when they bind to the apoenzyme, they will form a strong, tightly covalent bond. And the difference between these two things is that when the reaction proceeds and we have a product that is formed, what will happen is that let's say we is the coenzyme and wait, what happened? Oh. Okay. Yeah. Let's say we had an NAD here, okay? And it reacted and it became an NADH. So the thing with co-substrate is that when they react, they change their they become let's say NAD will change to NADH, and they will dissociate from the apo enzyme. Okay, they will dissociate from the enzyme itself and leaving the enzyme alone. While for prosthetic groups, because these are loosely bound, while for prosthetic groups, let's say for example, when we have a prosthetic group, an organic matter, for example, uh, let's say FAD, it will not dissociate. It will stay in its state, and it will go. It will for the apo enzyme and the co-substrate to go back, we need to return NADH plus to its original state so that it binds to the enzyme again and form a coenzyme for the enzyme to work. While for apo enzyme with prosthetic groups, you don't need to do that, okay? Because they will be tightly bonded to each other and they will not dissociate. That's the main difference between apo enzyme with prosthetic group and apo enzyme with coenzyme, okay, or co-substrate. Now we're going to talk about apo enzyme. Some enzymes are activated when they are bonded to metal ions such as iron, copper, and so on. So, wait. Okay. The quantification of an enzyme. Quantification of an enzyme is a measure of it is a type of measurement that we use to measure the amount of enzyme that is needed to convert the enzymes, the substrate, into product. This is called an enzyme assay. It is a type of measurement that we use in order to measure how many the amount of enzyme that we need to convert this substrate into product. So we have two units when measuring the enzyme's activity. We have the catal, and then we have the enzyme unit. The catal is the same amount of enzyme. The catal, it measures, it measures the amount of enzyme that is needed to convert 1 mole of substrate into product in 1 second. Okay, that's the difference. 1 mole of substrate into product in 1 second. While the enzyme unit, it measures the amount of substrate needed to convert 1 micromole of substrate into product in 1 minute. And we most likely use the enzyme unit because it's much easier to measure, because it's in 1 minute, not 1 second. It's like more practical. Now we have the isoenzymes. What do you mean by isoenzyme? Isoenzymes are enzymes that catalyze the same reaction, but the thing is that they catalyze the same reaction, but they come from different genes, and they are found in different types of tissue, but they catalyze the same reactions, okay? While isoforms, the difference between isoenzyme and isoforms is that isoform, they do catalyze the same reaction, but they have different kinetics, obviously, and different structure, but they also come from the same gene. The thing is, isoforms are mostly 90%, I think, or 99%, I'm not sure. They are not functionally important, unlike isoenzymes, which are important enzymes. So difference between isoforms and isoenzyme is that isoenzymes, they don't come from the same gene first. They are found in different tissues, and they catalyze the same reaction. Isoforms, they come from the same genes. Okay, so they're gene related. Now we're gonna go into the second part of the lecture, which is enzyme kinetics. So in enzyme kinetics, we have something called the Michaelis-Menten model. It measures how the reaction velocity depends on the substrate concentration when a fixed amount of enzyme is used. Okay, so it measures the reaction velocity, okay, between a substrate concentration and the enzyme. So it measures how many. Let's say, for example, we have a reaction of S converted substrate into product, and the enzyme that catalyzes this reaction is an enzyme E. Let's say, okay. So Michaelis-Menten measures the amount of enzyme needed to convert this amount of substrate into this amount of product over a period of time. The velocity at the beginning of the reaction is called the initial velocity. It is the velocity where this velocity measures the product formation at the beginning of the reaction, as soon as the product forms. How do we do this? We have two types of measuring the enzyme kinetic. We have continuous, discontinuous fashion, and continuous fashion. The discontinuous fashion involves mixing substrates and enzymes together and measuring the product formed after a certain period of time. So let's say I have this amount of enzyme and this amount of substrate. I mix them together and then I put them in the centrifuge. And then after, let's say, 10 seconds, I measure. After 20 seconds, I measure. This is what we mean by constant discontinuous method. While for continuous method, here, let's say you are studying the rate of the reaction over a long period of time. Let's say you add this amount of enzyme and this amount of substrate, you mix together, okay? And then you put in the centrifuge, and then you measure the rate of the reaction once from the beginning, literally you don't measure after 10 seconds, you measure it from the beginning till the end, okay? It's over a period of time. So how do we said we measure the enzyme, the rate of the reaction. How do we measure the rate of the reaction? How do we measure it? By using something called a chromogen, which is a substance that yields a brightly colored product. And this color product makes it easier to track the rate of the reaction. We will using a colorimeter or a spectrometer. So you use this chromogen, and then you track the dye, okay, or the color released from the product, and you see, now we're looking at the product formation, and this is how we measure the rate of the reaction. So this is a graph that associates with the rate of the reaction. Here we have the rate of the reaction velocity on the y-axis and the substrate concentration on the x-axis. This is a non-enzymatic graph that does not include enzymes, okay? It does not include enzyme. So you can see that as we increase the substrate concentration, the rate of the velocity increases because more substrate, more products, okay, formation, more collision and more product. While in enzymatic, you can see the difference between non-enzymatic and enzymatic. You can see that this here we have a plateau. So in an enzymatic reaction where we have enzymes involved, you can see that the slope is steeper. You can see that the slope is steeper here. Slope is steeper, and we have a plateau. Why do we have this plateau? Because here, as we increase the substrate concentration, we increase the amount of substrate that is bonded to the active site. But we reach something called the Vmax, where the enzyme would reach where all of the active sites are occupied, okay? And no matter how much substrate concentration you are increasing, it will not affect, or it will not matter, because we don't have empty active sites, so it will reach a plateau. Okay, Linus, Michaelis, and Maud Menton, these are the two scientists that discovered this equation, derived an equation to describe the initial reaction velocity as a function of substrate concentration. Okay, so in this equation, so you don't need to remember this equation, I'm just going to put it for your info. Okay, so the Michaelis-Menten equation generates a hyperbola graph. I'll show it to you in the next slide. Okay, here it says that the initial velocity, which is V0, equals to the Vmax times the substrate concentration divided by the substrate concentration plus the Km. So what do you mean by Vmax? Vmax, we said, is that it is when all the enzymes active sites are occupied, and Km is the amount of substrate needed to reach half Vmax. Okay, so how many substrates are you gonna need to reach half of the Vmax? Okay, is Km. It also measures the affinity of the enzyme for the substrate. Okay, so this is for Michaelis-Menten. Okay, now we're gonna talk about Linear-Weaversburg plot, where what it did, that it got this equation, and it took the reciprocal of the equation and generated this equation. 1 over V0, which is the initial velocity, is on the y-axis, and Km over Vmax is the slope. Okay, 1 over S is the x-axis, and 1 over Vmax is the y-intercept. Okay, 1 over Km is the x-intercept. So by the way, you need to know what does each of these stand for, because the doctor is going to ask you about it in the exam. He's going to tell you, for example, let's say 1 over Km was let's say 0.2, okay, and 1 over Km for the other enzyme was 0. Let's say 0.1. Which one is more? Which one has higher affinity, and so on. But I'll explain this in the upcoming slides. Okay, so we have isoenzymes. Isoenzymes, so we talked about them previously. Isoenzymes are two enzymes that have catalyze the same reaction, but they're coming from different genes, okay? And they are found in different tissues. This is an example of isoenzymes. We have glucokinase and hexokinase. The glucokinase is found in the liver, okay? While hexokinase is found in all tissues. Okay, you can see the difference in glucokinase and hexokinase in this graph. Okay, so this graph measures the glucose concentration over the enzyme activity. So glucokinase, you can see that glucokinase, the Vmax of glucokinase is much higher than the Vmax of hexokinase. And the Km, so we said the Km is half the Vmax. So you go to the Vmax, okay, on the y-axis, you go to half, and then you draw a parallel line parallel to the x-axis, and then you go down and you see that the Km for the glucokinase is higher than the Km for the hexokinase. Hexokinase has a Km of 0 and glucokinase has a Km of 10. What is the relationship now? How do we understand it? So we said Km measures the affinity, right? The higher the Km, the lower the affinity. Why? Because let's say this has a Km of 10. So for glucokinase, it needed a concentration of 10, a glucose concentration of 10 to reach half Bmax. While for hexokinase, it needed a concentration of 0 to reach half Bmax. Which one has more affinity? Hexokinase. So the higher the affinity, the higher the Km, the lower the affinity. And the lower the Km, the higher the affinity. It's inversely proportional. Okay? So the difference between hexokinase and glucokinase is that they have different Km and Vmax value, okay? So yes, glucokinase has a higher Vmax, but hexokinase has a lower Km, which means it has more affinity to glucose than glucokinase, okay? We have another example of enzyme is acetaldehyde dehydrogenase. This enzyme is used to break down the alcohol ethanol, okay? So we have two versions of acetaldehyde dehydrogenase. We have the low Km version and we have the high Km version. The low Km version, as we said, low Km, high affinity. It's very inversely proportional. And the high Km version has low affinity acetaldehyde dehydrogenase, okay? So what happens is that people who do not usually drink alcohol, when they drink alcohol, they're gonna have the high Km version because they never use it, okay? It's not that they never use it, they have the high Km version, okay? They're not used to alcohol, okay? So when they drink alcohol, this enzyme will have low affinity for the alcohol, and it will not break down the alcohol. The alcohol will circulate in the blood, and it will lead to facial flushing and rapid heartbeat symptoms after alcohol consumption, okay? While people with high Km version, let's say someone who always drinks and someone who never drinks, okay? When someone who always drinks takes a cup of, let's say, alcohol, he will not have these symptoms because he has the high affinity acetaldehyde dehydrogenase, which means that he will break down the alcohol much faster and more than the person who never drank alcohol before. Do you have any other questions? Is it clear? I think it's clear, yeah. So allosteric enzymes. Allosteric enzymes, so we talked about isoenzymes. Now we're going to talk about allosteric enzymes. Allosteric enzymes, they catalyze the committed step of the reaction. What do you mean by committed step? Committed step is the step that leads, and for example, A is the step that leads to this particular pathway, not any other pathways. For example, A can go to a path to another pathway, but this step is the committed step to this pathway. Okay. E1 here is the committed step in this pathway. Okay. The reaction for the allosteric enzyme will display a sigmoidal relationship to a substrate concentration on the graph. Okay, unlike this is the hyperbolic graph that I told you about for the mechanism. Okay, and this is the sigmoidal graph for the sigmoidal curve for the allosteric enzymes. Okay, so the committed step is catalyzed by allosteric enzyme, while the other steps are catalyzed by mechanism enzymes. So allosteric enzymes compromise several subunits that bind to substrate cooperatively. What do you mean by cooperatively? Cooperatively is that allosteric enzyme does not only have one active site for the substrate; it has multiple active sites for substrate. Okay. For example, let's take hemoglobin as an example. So when the first oxygen binds to hemoglobin, it has four subunits, four oxygen binding sites, right? So when the first oxygen binds to hemoglobin, it will find it very hard to bind to the hemoglobin. Okay, and then when it binds to hemoglobin, it will make it easier for the second one to bind to the second oxygen molecule to bind to the hemoglobin. Same thing here. When one substrate binds to the allosteric enzyme, it will alter the configuration of the other active sites in the allosteric enzyme and make it easier for the other substrates to bind to the enzyme. Okay. This is what I mean by cooperatively, okay? Allosteric enzymes are regulated by several activators and inhibitors. So the allosteric enzyme is regulated by two things: activators and inhibitors, okay? And these activators and inhibitors, they have their own site other than the active site, okay? Which is called the allosteric site, okay? So allosteric activators, which means they enhance it, they will shift. This is the normal curve, okay? Without the modulator. When we use an allosteric activator, we will shift the curve towards the left, okay? So we'll shift it upward and towards the left. While when we have an inhibitor, we'll shift it downward and towards the right, okay? So this is the allosteric enzyme, okay? Allosteric enzyme has two states. We have the T state and the R state. A T state means it's in its inactive state, and the R state it's in its active state. Okay, so when substrate binds to the allosteric enzyme, it will cause it to form. It will change it from a T state into an R state. Okay, which is the active state. So the regulation. How can we regulate allosteric enzymes? And the effectors bind at the regulatory subunits and affect the catalytic subunits. So we said that we have sites for the substrates and sites for the regulation, which are the activators and inhibitors. The activators or inhibitors, these regulators, they will bind on their side. Let's say the inhibitors, they will bind to the allosteric site, and they will change the configuration of the active site so that it does not bind active site of the substrate, so that it does not bind to the substrate, okay? And will stabilize the T state. As we said, the T state is where it is the inactive state of the enzyme, okay? The opposite happens for activators, where they will bind and they will change the configuration of the active sites and so that it facilitates the binding of the substrate, okay, and stabilizes the R state. So allosteric enzymes and kinetics. So as we said here, activators, what will they do? They will shift the curve towards the left. Inhibitors, they will shift the curve towards the right. And activators will shift it to the R state. Inhibitors will shift it to the T state, which has low activity. So this is another example of allosteric enzyme, such as the phosphofructokinase 1. This is an allosteric enzyme that is in glycolysis. Here, fructose 6-phosphate is converted to fructose 1,6-phosphate using fructokinase 1. Okay, ATP and citrate will feedback inhibit this reaction because they're going to be like, we have a lot of ATP, we don't want more ATP, so they will go and feedback on phosphofructokinase enzyme, okay, and will inhibit it. So they will bind to the allosteric site, and as we said, they will change it to the T. They will move it to the T state, the inactive state, and this is the normal curve. They will shift the curve towards the right. Okay, downward and towards the right. Okay, while AMP and fructose bisphosphate, they will activate or enhance this enzyme so that it moves the reaction forward. Okay, such as AMP and fructose bisphosphate. Okay, so let's say we use an inhibitor. So this is the normal. It went downward and to the left, and then we used AMP. Okay, and then AMP and or fructose 2,6-bisphosphate, they acted on the phosphofructokinase and they brought it upward towards the right. This is the graph for the activators, and this graph for the inhibitors. So as we said, so we were talking about feedback inhibition. This is the regulation of the reactions. Okay, so we can have the negative feedback inhibition, where the end product will go and feedback inhibit the committed step or the first step in the reaction, so that it's like we have a lot of the end product. It's telling us stop, we don't need more, so it goes back and negatively feedbacks on the reaction. Or we have the one for ATP and ATP. For example, here we have a lot of ATP, so the body is telling us we don't need more ATP, so the ATP will go and feedback inhibit the reaction. While if we have a lot of ATP, this means that we don't have a lot of ATP, so we need more ATP. So it's ATP will go and will positively feedback it will positively enhance the reaction. Okay, and then we have this one where the first product will positively feedback the reaction. I mean, positively feed forward. So forward feeding the reaction, and it will cause the reaction to move forward. So what happens if we said we regulate the allosteric enzyme by negatively and positively feedback, negative and positive regulation. Okay, what happens if we lose the allosteric regulation? So this is a patient of gout. Okay, here we have the enzyme called phosphorybasyl phosphate synthetase enzyme, which is an allosteric enzyme that is mutated. So when this allosteric enzyme is mutated, it is not regulated. Okay, what happens? This enzyme is part of the purine nucleotide synthesis pathway. It will keep on. So when it is mutated, and we have a loss of allosteric regulation, it will keep on producing a purine nucleotide, causes the overproduction of nucleotide, which will cause eventually gout. Okay, you guys are going to take this, I think, at the end of MOL, I think. So yeah, you just need to know there is a loss of allosteric control, which causes the overproduction of the purine nucleotides, and it formed a gout. Okay, enzyme inhibitors. So enzyme inhibitors are chemicals that reduce the rate of enzymatic reactions. Okay, they are specific, and they work at low concentrations. They block the enzyme, but they do not usually destroy it. So they don't destroy the enzyme. They just block the enzyme. Okay, and we have many types of poisons that work on the nervous system. Okay, so enzyme inhibitors. We have two types of enzyme inhibitors. We have irreversible and we have reversible. The irreversible inhibitors, they bind covalently and tightly to the enzyme, and they permanently affect it. So it's irreversible; it cannot be reversed. Okay, once they bind, they permanently affect the enzyme. Okay, the inhibition of this enzyme progresses with time, does not stop, progresses with time. Okay, an example is nerve gases and pesticides such as organophosphorus. Okay, will combine with serine residues in the enzyme of it will inhibit the enzyme acetylcholine esterase, okay, and this will block the transmission at the neuromuscular junction, okay. It's an irreversible block. The reversible inhibitors, we have three types of reversible inhibitors. We have the competitive inhibitors, we have the non-competitive inhibitors, and then we have the uncompetitive inhibitors, okay. So the competitive inhibitors, so as from its name, it's competitive inhibitors. So it competes with the substrate on the active site, and thus when it competes with the active site, with the substrate on the active site, what will it do? It will decrease the affinity of the enzyme to the substrate, okay. And this graph here, this is without the competitive inhibitor. With the competitive inhibitor, so as we said, it binds to the site of the active site, okay, and inhibits the substrate from binding, okay. But it is competitive inhibitor. It is reversible, so it can be reversed by what? By increasing the substrate concentration, okay. So it can be reversed by increasing the substrate concentration, okay. You can see here. So as we said, it decreases the affinity of the enzyme to the substrate. So what will happen? The Km will increase. So when Km increases, what will happen? Decrease in affinity. So this is a graph. This graph shows you that when we increase the amount of competitive inhibitor, the Km increases, okay. The Km increases, okay. So for competitive inhibitor, okay, the Vmax will not change. Why? Because if we increase the substrate, we will substrate will compete with the competitive inhibitor, and it will reverse the inhibition, okay. So the Vmax will not change, but the Km will increase. Which Km is going to be representing the competitive inhibitor? So these are all competitive inhibitors, but different concentration of inhibition, okay. But the one that is the least is the one that has more affinity. So let's say A1, which is the one that does not have inhibition, okay, is this one. This is the okay, this one is where we have this is the one without the competitive inhibitor, and this one is the one with the competitive inhibitor A2 with a concentration of 2. This is with a concentration of 4. So a question will come and tell you which one has more affinity. Which one will have more affinity? Obviously, the one without the competitive inhibitor. But with the comparing A4 and A2, which one will have more affinity? It's gonna be A2. Why? Because the Km is less than than A4. So the Km is less. So let's say a Km of 0.1 will be will have more affinity than a Km of 0.5. So the lower the Km, the more the affinity. Competitive inhibitor, as we said, is a structural analog to the substrate. So it competes with the substrate on the active site, and the inhibition, as we said, we can remove the inhibition by increasing the substrate concentration, okay. The Vmax does not change while the Km increases. Vmax does not change because we can reverse the inhibition by increasing the substrate. An example of competitive inhibitor is statins. Statins are used in hypercholesterolemia. When we have hypercholesterol in the blood, we want to inhibit the rate-limiting step, which is HMG-CoA reductase. So statin will bind to the active site of the HMG-CoA reductase, inhibit the formation of cholesterol, okay? This is an example of competitive inhibitor. So we finished from competitive inhibitor. Now the non-competitive inhibitor, here, the non-competitive inhibitor is different from the competitive inhibitor. The non-competitive inhibitor will not bind to the active site of the enzyme. Okay, it will bind to either the enzyme or the enzyme substrate complex. Okay, so when it does not bind to the active site of the enzyme, it will not alter the Km. So the Km will not be altered. The one that will be altered is the Vmax. So anything that binds to the active site of the enzyme will alter the Km. Anything that does not bind to the active site will not alter the Km, will alter the Vmax, okay? in the case of non-competitive inhibitor. Okay, so an example is lead poisoning and heavy metal poisoning. Okay, these are non-competitive inhibitors. So this curve for non-competitive inhibitor, as we said, it does not bind to the active site, so it will not have an effect on the Km, but it will have an effect on the Vmax. So it will decrease the Vmax because it will bind to the enzyme and the enzyme substrate complex, and it will decrease the amount of enzymes needed to bind to the substrate. Okay, so here you can see that it binds to the enzyme. Either it binds to the enzyme and form an enzyme substrate complex and leave the substrate alone. Okay, or it binds to the enzyme substrate complex and does not let the enzyme metabolize the substrate. Okay, so it will decrease the Vmax. So you can see the Vmax decreasing. Okay, can see, it's decreasing, it's changing, but the Km is the same. So it's not 100% the same, but it's similar. There's no significant change. Okay, so you have to know that in non-competitive inhibitor, it binds to the substrate, to the enzyme and the enzyme split complex, or the enzyme split complex, and there will be no effect on Km, but the Vmax will decrease. The uncompetitive inhibitor. So we talked about the competitive, the non-competitive. Now we're going to talk about the uncompetitive inhibitor. It binds to the enzyme substrate complex. It does not affect. So it is not affected. So it is not affected by the enzyme, the substrate concentration. I mean, why? Because it does not bind to the active site. So it is not associated with the active site. It binds to the enzyme substrate complex. Here, for uncompetitive inhibitor, it decreases both Km and Vmax. So it decreases both. It decreases the Km and decreases the Vmax. Okay. Why does it decrease the Km here? And why not only the Vmax? Same for the non-competitive, because here when it binds to the enzyme, so when it binds to the enzyme substrate complex, it will lock the enzyme with the substrate. Okay, and you're gonna have substrates that did not bind to the enzyme because you're not producing products. Okay, so more enzymes, so it will shift the equilibrium to produce more enzymes. So more enzymes will have more affinity for the substrates. So it will keep. So it will form more enzymes. So more enzymes are gonna be there to bind to the substrate, but it will only stay in this state. And it will only be enzyme substrate complex bounded to the inhibitor. That's it. That's why it decreases the Km. It causes increase in affinity of the substrate to the enzyme because we're gonna produce more enzymes. So for uncompetitive inhibitor, it's different. This is the graph for uncompetitive inhibitor. It decreases both Km and Vmax. Okay. So now we are done. I just want to wrap it up. So the uncompetitive and the non-competitive inhibitors, they do not get affected by the substrate concentration. Okay, it's only the uncompetitive inhibitor that decreases both Vmax and Km. Okay, these and the uncompetitive inhibitor is associated with the substrate concentration. So now we're gonna do questions. You guys can read this and answer the questions. So I'll answer with you guys. So here, the kinases are a class of enzymes that incorporate a phosphate into their substrates. The catalytic enzymes of kinases classify them as members of which of the following enzyme families. So here we talked about kinases. Kinases, they add phosphates. So which group of enzymes are these? They are transferases. So anything that adds a phosphate or kinases, it is related to transferases. Here, alcohol dehydrogenase requires oxidized nicotine amide adenine dinucleotide for catalytic activity. In the reaction catalyzed by ADH, an alcohol is oxidized to an aldehyde as an NAD plus is reduced to NADH and dissociates from the enzyme. The NAD plus is functioning as, here the answer is B, co-substrate, coenzyme co-substrate, because as we said, the co-substrates dissociate from the coenzyme. An example was NADH. So what does not dissociate is the coenzyme prosthetic group. It will not be the answer because they do not dissociate from it. And you are here, you're studying the effects of a new drug on the activity of your favorite enzyme. The results of your assay indicate that the drug acts as a non-competitive inhibitor, as we said. Which of the following changes to the Km and Vmax allowed the determination to be made regarding the drug actions? So we said non-competitive inhibitors. A non-competitive inhibitor associated with a decrease in what? The Km is unchanged, but the Vmax is decreased. Here, the drugs that inhibit the enzyme in a competitive manner. So we talked about competitive manner, which of the following effects on Michaelis-Menten equation derives parameters Vmax and Km? How will it affect it? Km will increase, Vmax will be unchanged. A sigmoidal plot of the initial velocity versus the substrate concentration. Which type of bond will it be? So a sigmoidal plot, we said it is associated with what? Cooperative binding, which is allosteric enzymes. So that is the end of the session. I hope you guys understood. And if you have any other questions, you can reach out to me. Thank you so much for watching, and you can scan the code for feedback.